Hi, everyone. Thank you for joining us today for the Exploring the Science of Acid Rain webinar. I'll be passing you off to our hosts, Colleen and Sarah, here in a moment, but I just want to go through a few housekeeping items. We will be monitoring questions and love to answer those throughout the session today. So feel free to use either the chat or the Q&A. We'll be monitoring both of those. And if we happen to not get to your question, we will follow up with you afterwards. And everyone who attends live today will also get a follow-up email with the link to the webinar recording, a certificate of attendance, and then I'll be sharing a link in the chat soon to some other resources that have the slide deck and the experiment files in a shared folder. So with that, I will pass it over to our hosts.
Hi, everybody. My name's Colleen McDaniel, and I'm going to start sharing my screen here. So thanks for joining us. Today, we're going to be talking about acid rain and kind of getting into the science behind it. So just today's agenda, I'm going to do an introduction of myself, and my colleague Sarah will be introducing herself as well. And we're going to give some background information about acid rain and then actually perform an experiment from biology with Vernier. And at the same time, we're going to demonstrate some awesome feature in Graphical Analysis Pro that allows you to actually share data over the internet. And then we'll be able to have some time for question and answers afterwards. But at any time, like Angie said, if you have any questions that come up or anything, don't hesitate to go ahead and type them into the Q&A section, and then we'll make sure that those all get answered. So a little bit about me. My name's Colleen. I am a biology and environmental science educational technology specialist here at Vernier. My background is I have a bachelor's in biology from Truman State University with a minor in chemistry. And I received my master's degree in science education from Montana State. Before I was a teacher, I was actually a research scientist looking at genetics. And once I decided I was tired of talking to fruit flies all the time, and I really wanted to get into education more to talk to actual students, I started teaching high school biology and AP environmental science in Houston, Texas. And after 6 years of that, I was lucky enough to get a job here at Vernier, where I get to do wonderful things like this, develop curriculum, develop sensors, and take your questions if you're ever giving us a call to ask us anything about our products. All right, now Sarah.
All right, my name is Sarah. I have a PhD in zoology from Oregon State University, and I also have some experience. I was a postdoc in neuroscience at the University of Texas at Austin, and I was also a professor of biology and taught for 15 years. So I taught everything from intro biology to human A&P, neurobiology, animal behavior, vertebrate zoology, endocrinology, so lots and lots of especially animal-related classes. And I also worked quite a bit with science education and K through 12 outreach, as well as doing a lot of professional development for my colleagues. And that is kind of what made me want to help teachers and help faculty in general to learn how to use more hands-on types of activities in the classroom. And that's why I love working with Vernier.
All right, thanks, Sarah. Okay, so one thing I just want to make sure is clear, we're here for you. So make sure you have any questions, don't hesitate to ask them. Sarah's going to be there answering them as we go. And at the end, we'll have time for other questions as well. And I'll make sure to kind of discuss them as we go. We are going to be demonstrating using Graphical Analysis Pro today. And if you have your own device that you have graphical analysis on, that's awesome. If not, go ahead and download it on your device. You can find downloads if it's a computer you're on from our website, or if you're using an iPad or an iPhone, you can download it from the App Store as well. If you already have it, just make sure you have it already up to date, and you do not need any of the sensors today, because what I'm going to be doing is actually sharing the data with you that I'll be collecting. So let's get some background information here. What exactly is acid rain? So rain is actually naturally in the more acidic range. On average, it has about a pH of 5.6. And the reason it's naturally acidic is this equation right here, where we have carbon dioxide in the atmosphere that's reacting with the water to form carbonic acid. So rain already has this kind of tendency to be a little bit more on the acidic side. What happens when we get acid rain is that you have components that are in the atmosphere that are actually causing the pH to be lower than that normal range. And I have that word deposition in there as well, because most often we're talking about just acid rain or moisture. It could be acid fog, it could be acid snow, it could be acid rain. It can be any sorts of moisture from the atmosphere coming down. But we also have some more solids and gases that can actually be deposited as well. So that's why generally when we're talking about acid, we're going to be talking acid deposition, which kind of is the all-encompassing term for both wet and dry acid deposition there. The two main culprits of this are going to be sulfur dioxide and nitrogen oxides that are found in the atmosphere. And that's these two equations here, where you have sulfur dioxide interacting with water to create sulfuric acid. And then you have our NOxes, which interact with water to create nitric acid. So these two compounds are going to be released by both natural sources and more anthropomorphic sources or anthropogenic sources. So for example, sulfur dioxide is most often going to be released when you're talking about coal burning power plants or other types of industrial plants. But it also is released when volcanoes erupt. So there is a natural process of this as well. And then on nitric oxides, you're going to have, those are going to be coming primarily from combustion of fossil fuels from vehicles, buses, things like that. And so what happens is these pollutants get released up into the atmosphere, where they then interact with water vapor, creating these acidic compounds, and then fall as most often wet deposition or dry deposition as in particles. Now, one thing I like to point out is acid rain is specifically a pollution driven problem here in the world. It is not related to climate change. That's ocean acidification, which is a different webinar that we'll be covering next month. So what are the effects of acid rain? Why is this bad? Well, one of the big things about acid rain is that it has a regional effect. It's not going to be a single point location where this is happening. So we see problems where, for example, we have high coal burning power plants that used to be very active in the eastern United States, and the emissions from there would actually travel northwards into Canada, resulting in acid rain deposition up there. So the big thing with acid rain is this regional effect, where this is something that can travel and have a much larger effect on a larger area than just where the pollution's actually coming from. And with the effects, we are going to see both direct and indirect effects of acid deposition. One of the most common ones that we're going to be looking at is the effect of on bodies of water. So we're going to look at what effect does acid rain have on different bodies of water and their ability to actually buffer that acid and help maintain a normal pH. You also have the effect that the acid rain with trees, it can actually do leaf damage and damage of different of their roots in the soil. One of the number one things we see in cities is building decay. So you can actually see that there are statues and buildings that have been dramatically affected by acid rain, especially those made of limestone or other soft stones, because the acid's actually causing them to deteriorate. And then with soil, you have the problem of, as the acid builds up in the soil, you can create some soil acidification, which can create an environment that is not healthy for microbes or plants growing in it. And some indirect problems we see are things such as metal leaching. So as the acid builds up in the soil, it creates an environment where certain ions are going to actually be released from the soil and leached out and either washed away, or they're going to have leaching out of harmful ions that can then be uptaken by plants, which can cause damage. The other indirect is disruption of food chains, where if we have an environment, for example, a body of water that's becoming more acidic, this can actually affect the phyto and zooplankton within that environment, which can affect the food chain, since we're usually talking about organisms that are lower in the food chain. So it can actually have some cumulative effects as we go. Okay, so the activity we're going to do today is from our Biology with Vernier book, and we're going to be using a Go Direct pH sensor. Now, a couple things to note, we actually have a couple different versions of this lab written up. We have a version in Earth Science, we have a version in the Chemistry with Vernier book, and within the Biology with Vernier book. So we have a lot of variety of ways to do this activity. We're going to be using the Go Direct pH, but you can use any other pH sensor as well. And one of the things to note when we're using this pH sensor is we're going to want to set it up in a little different way. Typically, our pH sensors are stored in a storage solution that has a pH of around 4. Most of this activity, though, we're going to be looking at pHs around the neutral range. So we want the pH sensor to kind of be preset at a pH 7. So for this experiment, we actually have the sensor that's been soaking in a pH 7 buffer solution for overnight, or just about 4 hours, to make sure it kind of gets in that neutral range. That way we see an actual reaction when we are adding things to the solution. So how this lab activity works is it starts with students just blowing their own carbon dioxide into water and measuring as the pH decreases because of the carbonic acid that's forming due to the CO2 output that they're giving it. From there, we are then going to measure the pH of different bodies of water. And these can be fresh water, salt water. They can also be different types of water samples. So for this experiment, I've actually created 2 samples, a hard water sample and a soft water sample. So we can kind of see how acid rain affects these differently, or if it affects them the same way. And our acid rain in this situation is we've created 0.05 molar sulfuric acid. So it's a very dilute acid that we're adding, and we're going to be putting in drop by drop. Okay, so let's get on with the demonstration now. I'm going to move our screen over. Okay, so here we have our graphical analysis. A little bigger here, okay. And so I'm going to switch over here to my little demonstration pod. First, I'm going to be putting on some protective equipment because I am working with acid. Even though it's a dilute acid, I'd still rather have goggles on just in case. And I have my pH sensor here sitting in my pH 7 buffer solution. And I'm going to press the power button to turn it on and select sensor data collection. Okay, find my pH, select that, and then done. So you can see I'm going to increase the size of my screen here so it makes it a little easier for everybody to see some of the values. There we go. And so we have a pH down here reading of 6.9, so it's around what I would expect in here. And for this first one, what I'm going to do is I'm actually going to go ahead and change my data collection settings. And instead of collecting for 180 seconds, I'm only going to collect for 30 seconds. Done. And so I'm going to take my pH sensor out and I'm going to rinse it with distilled water. Lightly dry it off. Put it in my ESUP. And then I'm putting it here. What I have is a beaker with 100 milliliters of distilled water. And just to kind of add to this demonstration, I'm going to put some bromothymol blue in this. Now, bromothymol blue is often the reagent we use in biology where we're doing like photosynthesis experiments and stuff. Well, bromothymol blue is actually an acid indicator. So what this is going to indicate is our our sample becoming more acidic because of carbon dioxide taking place. So a lot of times we don't think of it being that way. We kind of think of it as showing photosynthesis or cell respiration. And the reason I want to kind of show this as a demonstration here is to show how we can have something that's a fairly general indicator, but how nice it is to have a pH probe as well, so that we can get the exact measurements of a pH reading. So I'm going to put my pH probe in here, make sure the bulb is in the water. I am going to grab my high-tech straw and I am going to start my data collection and then start blowing, and we can see what's going to happen to our pH here. All right, so a couple things we can notice. First, my bromothymol blue turned a beautiful yellow color, showing that my pH did indeed drop based on the indicator. And that also on the graph here, we can see is that I did have a total change of pH and that it decreased. So what we have students do in this activity is they will then look at the beginning pH and then look at the final pH and subtract them to figure out what our total pH change was. So you can see we definitely had a pretty large decrease of our pH by blowing CO2 in. And that's where you have students start thinking about that chemical reaction of carbon dioxide combining with water to create carbonic acid. Are there any questions so far?
Colleen, there's nothing in the chat yet or the QA.
All right, perfect. So hopefully that means I'm explaining everything beautifully and everybody understands. Okay, so for the next part of this lab, what we're going to do is start with a hard water sample. And I'm just going to pour about 100 milliliters into my beaker here. And for this part of the activity, you can just swirl the beaker, but because I'm working by myself, I'm going to add this fancy little thing we have, which is a little magnetic stir thing, flapper that actually attaches to the bottom of the pH probe and allows you to stir it on a magnetic stir plate. So I'm going to go ahead and put that in. All right. Now, to do this next activity, what we're going to be doing is we're going to be adding drops of dilute sulfuric acid or acid rain and measuring how much the pH changes with each drop. So since we're not doing something time-based, what I'm going to do is do a new experiment. I'm not going to save. Still do sensor data collection. So it's going to kind of restart what I have going here. And in my mode, I'm going to change from time-based to event-based. And I'm going to do events with entry. And so my event name is going to be every drop. And my drops do not have units. So I'm not going to worry about that. And then I'm going to select done. And so you can see things change where now I have a data table along with my graph here. And before I start, where it says data set 1 up here, I'm actually going to rename this data set and call it my hard water. That way I know exactly what I'm measuring here. What is my sample? The other thing is this meter down here is a little small. So I'm going to turn on a bigger meter so that I can actually be monitoring this pH as I go. Now, what's wonderful is I can just press collect. And since this isn't time-based, I don't have to worry about rushing and getting this experiment going. So I'm going to go ahead and turn on my little stir and start agitating the water. And my first sample I'm going to be collecting is 0. So I'm not going to add any drops. So I'm going to press keep and enter 0 and keep point. Now I'm going to add 1 drop of my acid rain. And I'm going to let it sit for a little bit to make sure that acid amount has kind of been able to work its way and get dissolved into our solution here. Sorry if this is a little squeaky sounding. All right. So I'm going to go ahead and press keep now. And I've added 1 drop. Okay, so now I'm going to add another single drop. And now the total number of drops I've added is 2. So again, I'm going to let that kind of sit and let it dissolve. And sorry about the squeaky noise, if you can all hear that. Hopefully you can't. Okay, so I'm going to keep it now again. So now that's 2 drops. I'm going to add another drop. And I'm going to keep doing this. I'm going to eventually add a total of 10 drops to my sample here. So it's going to be very exciting for a little while. Okay, it looks like it's kind of stabilized around there. I'm going to keep. Now that's my 3rd drop. I'm going to add my 4th drop. Okay. 4 drops. Fifth drop. Okay. Now drop number 6. Drop 7. Drop 8. Drop 9. And drop 10. Okay, so that's my first set of data here. And so when we just look at it like this, it's going to look very much like a straight line because the amount of pH decreasing is a very small amount. So if you think about trying to do something like this with bromothymol blue, it'd be pretty much impossible to see a gradual change. It would either be basically green, blue, or yellow. So the pH probe is going to be able to help us pick up these specific things. Now I'm going to switch out my water sample and go from a hard water to a soft water. Give everything a nice rinse with some distilled water. And then add in some soft water. Okay. Drop that in. Okay. And we're going to press collect again because we're going to be doing a new data set now. And I'll rename my data set after we've started. So kind of the same process here. I'm going to go ahead and keep my first point, which is going to be 0 drops. And now I'm going to add 1 drop of sulfuric acid, of my acid rain or dilute sulfuric acid. You can see this one's changing a lot faster than the last one. And keep that. And drop 2. Drop 3. Drop 4. And you might be wondering why I'm choosing to press keep at certain times. And it's basically how I'm doing this. And this is going to be a problem with students, is they don't know when the number is stable enough to press keep. So I'm watching my live readout here, and I'm seeing that's kind of going from 6.96, 6.95, 0.94, 0.93, and now we're hovering between 0.92, 0.91, 0.93. And so basically what you're looking for is the values to stop moving in a specific direction, in this case, downwards. So you guys can see now it's kind of hovering about 0.91, 0.92. And to me, that means we're kind of at our reading. So I'm going to go ahead and press keep. Oh, as we keep going down a little bit more. And it doesn't have to be super precise that when you're pressing keep. It's a matter of making sure you're getting that overall trend. And that can be something that's really difficult for students to understand or be comfortable with, because they always have this idea of they have to do it perfect, they have to do it right, and they're worried about messing it up. What's the nice thing about this experiment is that either way, you're going to get that general trend downwards, even if you accidentally press keep too soon. We'll still see this kind of eventual decrease as it's going down. And so once I start seeing the numbers fluctuate back and forth between a high and a low, high and a low, that's when I'm going to go ahead and press keep. 5 drops. Put in my 6th drop. Seven. Eight. Nine. and 10.
Colleen, this is just a comment, not a question, but those are beautiful data. The difference between soft water and hard water is really remarkable.
Thank you. That's what I like to see work. It shows me I did this right. Okay, so that is our last data set, and I'm going to turn off the stir so we don't have to keep listening to that. And then now you can see my beautiful face instead of the thing. So I'm going to go back, and like I renamed earlier, I'm going to go ahead and rename this data set. And this is going to be soft water. And now what I can do is I can pull up both my data sets together and take a look at them. So if I click over here, I'm going to go ahead and have pH shown for both hard water and soft water. And you can see there's quite a dramatic difference between these two samples I did. So what does this tell me about the effect of acid rain on hard water versus soft water? That is a question somebody could answer. So what we're seeing here is that hard water actually is going to have more buffering ions available in it than soft water does. So hard water is going to be in a situation where, because it has more of these buffering ions in it, as acid is added to it, it's not going to change as much. It's going to be a very, very slight change. While in soft water, you don't have as many of those. So you're going to actually see the pH is going to get lower and be affected more dramatically by the addition of acid to the environment. So this is something that a lot of students don't really think about, because usually we're concentrated very much on just our little region. But that could be something that could be pretty cool, is if you're able to get different water samples from different areas around you and test the buffering capacity of each of these samples and kind of talk about health of waterways and what this could actually mean for that environment. Now, one of the cool things we can do here is by activating Graphical Analysis Pro. So I am going to stop sharing, and I'm going to share my presentation again. And so what we're going to do next is we're going to share my data to y'all. So with Graphical Analysis Pro, which basically is a pro feature that you unlock in regular graphical analysis, we have the ability to actually share data over the internet. So when you launch graphical analysis, there is going to be a place that you can enter a code to activate the pro features. So Sarah, could you put this code into the chat window so people can add that and activate their pro features? So this is where,
give me just a second to type it in.
This gets a little weird, because what we're going to be giving you are two codes. So there's going to be one code to activate Graphical Analysis Pro, and this is going to give you 2-week free access to the pro features. And then I'm going to give you a different code once you've activated it, so that you can actually do data sharing and see the data I collected, and then actually have your own set of this data.
All right, it is in the chat.
Perfect. So use the code from the chat, and then I'm going to now go back to graphical analysis. And once you have activated that, where you're going to want to go is where it says sensor collection, data sharing, manual entry. You're going to select data share. And I'm going to turn on a data sharing session. And this is the code for the data sharing session that you'll want to enter.
I'll put that one in the chat as well.
It is kind of useful that they're different lengths, so it kind of helps.
And that is a 0, not an O.
Yes.
All right, if anybody wants to cut and paste it from the chat, it's just that little 6-digit code.
And so how this works is every data sharing session is going to create a unique new code. So anytime you start this, it'll be a brand new code. You're not going to have to use the same code over and over again. While Graphical Analysis Pro, you have a code that you basically just activate it once, and that's all you have to do. It'll be good for as long as the activation code is good for. So this here, when I exit out of this, I can see I have a little green dot over here in my data sharing. That shows that I am actively sharing data right now. And if I click on it again, it actually shows me that I only have one person currently connected.
That's me.
And that's Sarah. So this will give you an idea of how many people are connected and looking at this. Now, the reason that this is kind of a nice feature is, barring COVID, if you are having students that are missing class or lab activities, they can actually still participate while at home if they have internet connection, and their lab group could actually just share the data to them. And the key thing here is that I'm able to actually do my own analysis. So I can show my statistics of this activity here. And if you're looking at the data sharing, this statistics box does not show up on your screen. So each student is going to be individually responsible for doing their own data analysis on here, which is kind of a nice feature, I think, instead of having one student be doing all the work for everybody else. And what we're going to have students do in this lab activity is the same thing we did for the first one, is we're looking at the total change in pH. How much did that pH change, and what all does that actually mean? So I only did 2 water samples, and you can see I was able to do 2 water samples within about a period of maybe 10 minutes. You are able to do more than just 2. And like I said, a lot of times people will do, for example, you can do fresh water versus salt water. And you can do it as well so that that students are sampling different waters from different lakes or ponds within an area. You also have the ability to just make your own water. So the soft water and hard water that I have here is actually a homemade variety, where all I did was take distilled water, and with the hard water, I mixed in about so this is 200 mils of hard water. I put in 20 milliliters of a pH 7 buffer. So that's adding basically enough ions to this to make it a nice hard water sample. And then to make the soft water, I added 20 milliliters of my hard water sample to 180 milliliters of distilled water to make 200 mils total solution. So the soft and hard water, you can actually make your own. You don't have to worry about going out and finding different water samples and things like that. So you have the ability to do that. A lot of times when I'm traveling, and well, when I used to travel and go to conferences and do shows, I would actually just make my own water from scratch. And I would add different buffer solutions or add some sugar or some salt and basically different things just to kind of make a mucky mess and see what I could get out of it. So are there any other questions right now? So once students are done collecting their data, they actually have the ability to export the graph, and they can export it as a graph image so that they can drop it into any sort of lab document they might be doing. They also can here actually add a graph legend, so we can actually see on the graph what specific hard water and soft water, what the colors represent. I also have the ability where I can do some graph options where I can add a title here of hard versus soft water, or we could do water buffering. Something like that. So you have a lot of ability to kind of modify and goof around in here. And this program is going to run the same whether the students have Chromebooks, iPads, computers, doesn't matter. It all is going to look the same and run the same on each one of those. So I'm going to wrap up here. So some extensions that you can do after this lab is we do have another one that's looking at the effect of acid rain on soil. So you, instead of looking at water's ability to buffer, you can actually look at the ability of soil being able to buffer and how different soil types and how you can maybe modify those soil types to increase their buffering capacity. If you're talking about, say, buildings and building structure and the effects of acid rain on those, you can actually look at the weathering, where you could have like limestone, marble, basalt, and granite sitting in different solutions of sulfuric acid, different and low concentrations, and see what actually happens to each of those as they've been sitting in there. And kind of a fun extension engineering way is just to have students think of ways and test ways that they can actually create buffering solutions to some of these different habitats to help improve the quality of them and improve the resiliency of them against different types of acid deposition. Overall, the good news is in the United States, because of a lot of the Clean Air Act rules that were implemented, we do not have as big of a problem with acid rain as other areas of the world do now. But it's still an important topic for students to understand, especially when we're discussing pollution and the effects it has on both the environment and on people in cities. So that's our presentation today. Thank you all for coming in and joining us. If you have any remaining questions, please go ahead and ask them. If you have any questions afterwards, feel free to email us support@vernier.com or the biology team, biology@vernier.com. We're all here to help and make sure we can do whatever we can to help improve things for you all.
Colleen, we do have one question. Where in the world is acid rain the most serious?
Oh, that's an excellent question. Right now, mostly in China is going to be the area that is going to have the most effect. There are areas in Northern Europe that have kind of some long-standing problems just due to historic use of coal as a power source. But the main area you're going to be seeing the biggest effect is in Asia. And specifically, there's concerns of some of those acid particles actually being brought into the atmosphere over here to the western side of the United States. So that's kind of the thing I like to emphasize the most with acid deposition is that it is not necessarily a local problem. It's a regional issue where it happening in one location can affect many other locations that are miles away. And yes, it's much more global than just certain specific areas. Any other questions? Thank you, Maureen. I'm always happy when the data actually turns out so nice.
Those were some pretty awesome data.
Woohoo! I made my solutions well. The partial chemist in me is very happy. Okay, so yeah, the district has a limit. So Logger Pro is what I recommend really is looking at graphical analysis instead of Logger Pro. We're finding that depending on the age of the computers, especially if the students have Chromebooks, graphical analysis is actually a lot easier to put on them and then Logger Pro is. And it's primarily because Logger Pro is a computer program, while graphical analysis is an application. We are happy to talk to district IT people, and that a lot of them can get that, and we can make sure that we have all the permissions necessary for them to be put on their computer. Oh, excellent. So I see Betsy asked a great question. Can you export the data to Excel or Sheets? Yes. So the data can be exported as a CSV file, so comma separated values, which can then be opened in Excel or Google Sheets. There is some analysis on here I wasn't able to go over because it didn't quite fit in with this lab, but you are able to do statistical analysis and different curve fits on the data. This lab, we don't do curve fits because it's not necessarily a linear relationship what we're looking at. So, but you do have the ability to do curve fits on data that is called for that.
Oh, question about when our next presentations are.
No problem. Let me.
You want to take them to the website?
Yep. I am going to take you to vernier.com. There we are. So if you go under here to educator training and over to webinars, this will give you a great list of the different webinars that we are going to be doing. So if you scroll down, this one we just did, Exploring the Science of Acid Rain. We've got a bunch of different topics for different subject areas. And my next one I'm going to be doing is Shedding Light on Ocean Acidification. And that's going to be right before Thanksgiving on November 23rd. And all of these will also be recorded. You can also, when you scroll down here, it allows you to browse past webinars. So you can go into our past webinars here and actually look at specific topics that you might be interested in. So you can actually go by subject and isolate just the biology ones or earth science or things like that.
Yeah, so we have a comment that the times are when we're in class. That's exactly why we put them on as recordings after the fact for anybody that happens to miss the live presentation. We do have them available as a recording for any time that you'd like to watch it.
Mm-hmm. And if you have suggestions on how we can make it work better for you, please email us and let us know. We're always open to suggestions because we want to make sure that what we're doing is going to be effective and useful for you, because that's what we're here for. We want to make sure you're going to be successful doing these different activities. Or if there's a specific activity you would like us to do, send us a message on that too. And we're happy to always take those. All right. Thank you all for joining us this afternoon. And I hope you all have a wonderful day and a great rest of the week. And sooner than you know it, it's going to be winter break. Have a great day, everyone. Thank you.